Swayam Prabha. Digital India. Educated India.
All right, so we'll start with this next lecture. Again, let's see how interactive I can keep this particular one at least. So the idea here is to give you an overview of the topic that I work on and what I, as you pointed out very nicely, was my interests are in understanding how the brain functions. What is your name? Pranav, as Pranav pointed out. Okay, so my interests are in understanding how the brain functions, and the way I aim or I approach it is by looking at the neuronal circuitry. So the idea is that whatever behavior or whatever the output of the brain is, the computations that are performed by this circuitry, how they function, that underlies whatever behavior or whatever processing that the brain does. So you can think of the brain as a holistic organ, but as you go deeper and deeper into it, if you zoom into it, at the bottom line, the unit of any brain system are these neuronal circuits. Okay, neuronal circuits are basically neurons which are connected to each other. Yes? Huh?
What is the most interesting? We see that seizures are caused due to excess electrical signals in the neurons. Have you found anything interesting related to that?
So that's a very broad question. So I do not work on seizures. But if you look at the field, seizures are caused because of dysfunction in neurons. Basically, what happens is these neurons start to get activated a lot more. Okay, and in order to control their activity, there are usually other neurons, and those neurons start to dysfunction. And so what ends up happening is there is hyperactivity in neurons. Now, the hyperactivity is caused by multiple mechanisms. So the problem is, if you have 100 patients, the effect is the same. They all have hyperactivity in neurons, but the reason it was caused is different. And so we are still struggling with understanding the reasons that are causing it. So nowadays, the best and this is true for most of neurological disorders. We end up controlling the symptoms. So the symptom here is hyperactivity in neurons. It is not the cause, right? So what we do is we control the symptoms. But obviously, the original cause is still not controlled. We are not combating that. And so the disease usually ends up progressing to a worse stage. We don't have mechanisms to combat. And this is true for multiple neurological disorders. We have mechanisms to reduce the effects, but over time, the effects start to become worse. And that's because we do not understand the mechanisms of what happens at the level of these circuits. Okay.
You also work on stuff like brain-computer interface.
I don't. But what is your question?
Do you think that will be able to help us understand these processes?
Okay, so what is a brain-computer interface?
So that I just heard about Neuronal, so I heard that is brain-computer interface.
No, but what do you think that means?
So it is able to sense the signals in neuronal circuits and accordingly, predict the actions.
Okay.
Or the outputs of.
Okay, so now what is your question?
Yes, sir. So I just wanted to know if you work on it.
Okay, no, I don't work on it. But the idea, again, is that you need to understand how neurons work so that you can interface it with a system. I work in understanding how neurons work. I do not work on the application end of it. But there are people in this institute who do work on it. Okay. Okay, so again, I am interested in understanding how these circuits work. What do they actually do? Now, if you look, this is from Bangalore. Bangalore people may know. Okay? Yes, Bangalore traffic is horrible. Worse than Chennai, I'm going to say straight up. All right, so if you look at this scene, you can see this is what our visual system, if you are driving in this traffic, this is what our visual system deals with, okay? Look at the plethora of stimuli it has to deal with. You have big buses that are moving slowly. You're going to have two-wheelers who obviously are going to go at high speeds, zipping by. You also have people crossing the road at the wrong place. So you have to be aware of them, and you need to know at what speed they are moving. Not only are some people moving, but some people are talking, so you do not know when they're going to start to move. So there's multiple stimuli that is involved. Our brain is hit with all these stimuli at the same time. You have multiple objects, multiple speeds, moving at different direction. So your brain has to process all this information, okay? And the way it has to do it is it has to faithfully look at each object. It has to understand which direction it is going to move, and then it has to predict where it is going to be so that you can avoid all the obstacles and then go safely. And obviously you're not thinking about it. If you're driving, I guess you are not ready to drive yet, right? Okay, when you will be ready to drive, I'm sure you've all been in a car. Clearly, accidents are not as common as you would think it should be, given the variety of stimuli that you're dealing with. Now, not only do you get a variety of stimuli, you have to process all this in a millisecond, right? In a split. It's not really millisecond, but in hundreds of milliseconds. Our reaction, do you guys know what is our reaction time? And so it's actually about 150, you can think, at the fastest, okay? And so you should not only, so the way it works is the system senses the stimuli, then it processes it, then it goes to the motor system, which actually has some output to it, okay? So there's three arcs. One is it senses, then it processes it, and then it has to do an output. And all of that happens within 200 milliseconds. Very rapid. So how does the system manage it? When you give it so much stimuli, as you can imagine, computers and where we are, the technology is really slow if you have to navigate through all these systems, right? It's not as effective as it should be. And that's because we have still not reached a stage where we can replicate what the brain does. And I'll give you a glimpse of why what the brain does is so impressive that it's not easy to replicate. Okay, so but the bottom line is you have a variety of stimuli which has to be processed in milliseconds, all right? So how does the brain do it? So the way it does it is it takes this whole composite scene that we are seeing and it breaks it down into parallel channels. So one channel will likely process, say, motion. One channel will likely process color. One channel will likely process the size of the object. So they are not all doing the same thing. They are doing parallel work. And then higher in the brain, they get to recombine. Then it sees, oh, we have an object. I have an object which is moving in that direction. Now I want to know what is the size of the object. Then it recombines the information at higher in the brain. Then I want to know what is the color of the object. I want to know perhaps sometimes what is the sound the object is making. Okay, so those are all important factors. So what happens is that the information gets decomposed into multiple streams, okay, with each stream picking something unique from the visual scene. And then higher in the brain, depending on the decision that needs to be made, different streams end up recombining. All right, so this is a basic principle that you see in neuroscience in how the way brain works. It decomposes and then again recombines the information higher, depending on what it wants. Yes?
How do we know this actually happens in the brain?
Okay, let me give you some examples. Okay, so your question is, how do we know the information is decomposed into different streams?
Yes. Is there a set number of streams or the type of streams the information is being decomposed into, or does it change from?
Okay, so it depends on the part of the brain you're at. If you're in the early stages of processing, and from now onwards we will only talk of vision because that is the best studied and that is what I study. Okay, so most likely whatever questions you have, I will be able to answer better. But think about the nervous system as a module which replicates itself. What happens in vision? does not necessarily, is not restricted to that. The way information is processed in vision is similar to the way information is processed in sense, in touch, the way it is processed in other aspects. Okay, so whatever we discuss now, you can think of it as a general principle of how the nervous system in general, how the nervous system works. So yes, there is evidence, and we will see in a slide or two on how many streams like this is possible where the information gets decomposed. Okay, we do not understand all of this, because if we do, then we will be able to recreate a brain already. Okay, but the bottom line is it decomposes the information. Okay, so we will start with one example, and this is the example that I will be showing you is the retina. You guys know where the retina is? Okay, so it's in the back of our eyeball, and it is a very thin piece of tissue. It's about the size of a coin, and it's very thin. The thickness is about 2 A4 sheets. That's it. Now it has millions of neurons, compact neurons in there, and it has the ability to obviously process all the information that all of you are seeing right now in front of you. Okay, so you're looking at two really small sensors, but the amount of information that is processed there is sufficient for you to reconstruct the whole image in our brain. Now bear in mind that what the retina sees is not actually what the brain paints for us. In fact, the image that falls on the retina is inverted. And so, but obviously I'm not seeing you all inverted. In fact, if you put glasses that inverts the image, right? Initially you will see it inverted, but over time our brain adapts to that change and realizes that something is wrong with the image that I'm seeing, so it ends up re-inverting that. Okay, so again, remember whatever our sensory system sees is not necessarily what we perceive. Okay, that aside, whatever information goes into our system goes through the sensory system, and we start with the retina. If you look, so this is just a cross section of the eye, and this red layer that you see here lining the back is the retina. And if you look at a cross section of the retina, you see this pretty picture. This is a stained cross section. So the neurons that you see on top, green here, these are the photoreceptors. They are the ones that when light falls on it, they convert the light, transform the light, transduce the light into electrical signals. From the photoreceptors, the signals go to the next layer here. So the green cells that you can see here, so this is where the cell body is, the soma as you call. Now if you look at neurons, do you know what neurons have? What is the typical parts of a neuron? Axon, dendrite, and cell body. Okay, some of them have myelin sheath, yes, but the basic structure is you have a cell body, you have dendrites, and you have axon. So this is a really small neuron. This must be about 20, maybe 40 microns at best. So this is the cell body of this neuron, and you can see this thin line going up. So that is actually the dendrites. And you can see another thin line going down here. That is the axon. And you can see these structures here of the green cell. That is the terminals where the neuron is releasing its neurotransmitters. So this is called a bipolar cell because it has only two structures, neurites, that are coming out of it. One is the dendrites and the other is the axon. So this would be the photoreceptor. Signal from the photoreceptor is transmitted to the bipolar cell, and the signal from the bipolar cell gets transmitted to the cells in this layer. The third one here, none of them are labeled here, but you can see these blue structures. So that would be the nucleus of ganglion cells. Ganglion cells are the output neurons in the retina. So whatever information is sent to higher centers in the brain, it has to happen at the level of the ganglion cell. So if you try to understand what is happening in ganglion cells, we can try to understand, we will know what is the information that is being sent to the brain. How the brain processes it is another question in itself. In this case, what I'm going to show you is how we can study how ganglion cells process information. Okay, so that we will keep it to a very small aspect of a much larger picture. So you can also see some other cells. So you can see this magenta cell here. You can see this in magenta here, which has, so this is the soma here, and then it has these long lines here. So those are called amacrine cells. So there are 5 major types of neurons in the retina. You have the photoreceptor, which is the main, which when light falls on it, it converts into electrical signals. And then you have the bipolar cells, and then you have the ganglion cells. And whatever information is sent to higher centers in the brain goes through the ganglion cell. Okay. We also have 2 other types of cells. I've sort of drawn one here. This is called the horizontal cell. And then we also have the amacrine cells, both of which are present in the retina, but their job is to shape the signal that is being sent to the ganglion cell. Okay. So without the bipolar cells or the photoreceptors, there will be no signaling in the retina. Okay, so they are the major neurons. So photoreceptors excite bipolar cells will signal to the ganglion cell, and ganglion cells will send the information to higher in the brain. Yes? Shaping.
What does shaping the signal mean?
We will see. Okay, yeah. So again, what I want to, the main point here is if you were to remove the photoreceptor, there will be no signaling in the retina because you don't have the ability to transform light into electrical signals. And this is a disease that is, and many of the common visual disorders have this problem where the photoreceptors are affected. Something called as retinitis pigmentosa or age-related macular degeneration are cases where the photoreceptor is degenerated, and so the tissue is not light sensitive anymore. And again, the same is true for bipolar cells. If you kill the bipolar cells, the signal will not reach the ganglion cell. Photoreceptors will be working, ganglion cells will be working, but because the bipolar cells are not there, you do not have a connection between these two. And there are diseases where bipolar cells are affected, and as a consequence, vision is affected. So every disease that you see, some part of these neurons are likely to be affected. Now, the amacrine cells and the horizontal cells, if you kill them, what happens is there will still be signaling, but certain aspects of signaling will be lost. We will see one such example if time permits. Okay. Now, although I said there are 5 major types, right? If you look deeper or zoom in, each of these types are multiple subtypes, okay? And in the level of the mouse, for example, we know as of now the mouse retina is the best studied, and we know there are 14 types of bipolar cells, we have 60 types of amacrine cells, 50 types of ganglion cells. So to answer the question that was asked, how many channels are there, right? So the number of channels will depend on the number of ganglion cells. If there are 50 types of ganglion cells, you can have 50 different types of information that is being sent to the brain, okay? So the latest count, there are 130 types of neurons in the mouse retina. In the human retina, as of now, we have about 70 is the latest count. But we are not there yet in terms of techniques. These numbers are likely to go up, okay? So the bottom line is, we do not have a tissue which is made up of cells of the same type. We have a tissue which is made up of 130 types of cells, okay? So how does this look? So you can think of these as blocks. You can think of these as Lego blocks, each cell. And if you were to put them together, you can end up getting very different structures. You can get up very different outputs. And that is what the system does. The system, it gets inputs from different types of neurons, and then you end up getting some really impressive computations. Okay, so this is the bottom line. You have 100 types of neurons, and they can be connected in different ways. So you can think of them as Lego blocks, and then when you put them together, you end up getting a circuit that can give you some really impressive computations. We'll look at one computation. Yes? Okay, so if you go back to this picture, right? So there are multiple ways in which we are able to find out. One is the kind of molecules neurons express. So different types of neurons will express different types of molecules. So if you see here, the green is a molecule that is expressed only in these cells. They're not expressed in all the cells, right? So likewise, if you look at this magenta color, that is again labeled for a molecule. And that molecule is expressed only in this type of cell, not in the other types of cells. So that is one way. The other way, which I will show you, is by electrophysiology, where you record from these cells, and then we see what kind of responses they give. That is the ultimate way, because all of this is just indicative, but to look at the function, we have to record from electrical signals of these neurons, and we see that there's at least 30 different types of signals that you see at the level of the ganglion cell. Okay, so but if you look at the basic blueprint, you have the photoreceptor, bipolar cell, and ganglion cell, and there are also amacrine cell. But if you look at the subtypes, you can get a variety of circuits. Okay? Okay, so what I'm going to show you now is a simulation, not of the retina, but of the visual, of the somatosensory cortex. What you're going to look at, so to just give you an idea of, remember, I told you there are 100 different types of neurons, but they are all embedded in this one tissue. So the way this works is you have 50 to 100 different types of circuits, and they're all present in the same tissue. They're all processing information parallelly. So what that looks like is something like this. Can you guys see this, the video? Okay, that one looks better. Can you guys see that? Okay, so wait, I should pause this. Okay, so let me orient you. So what you're seeing here in blue are the neurons. When the activity of a neuron increases, the color changes. Red means red or yellow. Yellow is the maximum activity. Red means there is some activity. Blue means there is no activity. There's just a neuron. Okay. So what is being shown here is a very small part of the somatosensory cortex of the juvenile rat. There's supposed to be about 10,000 neurons in this simulation. Now, as I said, the whole retina has millions of neurons. Whole cortex will have millions of neurons, billions of connections. So that's very difficult for us to, as of now, model. But the point I want you to see is that you will have many neurons, but they're all not going to be active at the same time. They're going to be active at different points of time. Sometimes they will all get active. Sometimes some of them will be active, indicating that they're all doing something different. They're all adjacent to each other. They're going to be next to each other, but they are different types. So they are doing something different from each other. So that's an indication that that was active. So now you can see that these neurons are active. This one is not active. So this is just getting zoomed out now. And as you zoom out, you get an idea of the scale of things. And again, remember, what you're seeing is maybe 1% or less than that of the cortex. Okay. So as you can see, sometimes all the neurons are going together. Sometimes one of them is going on. Sometimes 2, 3 will go on, because they're all different types. They're doing different things. Okay, the information is being processed. So now comes the question that some of you have already asked. Okay, I have this network, and I'm telling you, and I have been telling you, well, we have neurons which do different things, and this is a simulation. How will we go about studying these neurons, right? So how do we do this? So the way we do it is by something called as patch clamp recordings. Basically what we do is we put a glass electrode, we have a very small tip. The tip is small enough that you can go and patch on to one cell, and then you can record the electrical activity of only that cell without interference from the surrounding cells. Okay, so that is the technique, that is the golden standard for understanding what one neuron's activity looks like. Yes? Huh? The clip. It's not a clip, it's a tip. This tip, right? So this is an open hollow electrode, glass, and it is small enough. I will show you a video. It is small enough that it is likely to be a micron. Yes, micron or less. Usually microns. So that is actually an easy task, because this has been happening for 50 years now. So now we have machines where we can pull pipettes of the exact type tip that you need. Okay, so that's pretty standardized now. This technique has been around for a while, but this is still sort of like the golden standard. So basically the way it works is it hits up the glass and then it pulls the glass, and it happens in a controlled manner, controlled enough that you can get reliably the same tip size. Okay. So this is what the setup would sort of look like. So because you're looking at single cells, you have to use a microscope. Okay, and so this would be the objective. So you put the tissue in here, and you see this thin, long line. So that would be the electrode. Okay, so that would end up tapering down to the size which is smaller than a cell. So this is yes. Yes, they are live cells. If they are not live, then we do not know how their function is, right? They will not be able to communicate. Do you guys know how neurons communicate with each other? But what is... Huh? What is neurotransmission? They do... Well, can they send electrical pulses to each other?
If the synapses are very close...
What is a synapse?
The joining between an axon and a dendrite.
Okay, but what, I mean, that's one. Two cells, there is going to be gaps, right? What is special about this? Fluid. No, it's not a fluid, man. No, there are chemicals called.
Chemicals called neurotransmitters that are released by one.
Yes, so what happens is, in terms of communication, one neuron releases chemicals called neurotransmitters, and the release of neurotransmitters is dependent on the electrical signal, and that chemical goes and binds to proteins in the next downstream neuron, and that triggers electrical signals again. Okay, I don't know why I started this question.
Firstly, we have so many neurons together. The fact that they all operate at sometimes the same time, and the fact that they even use neurotransmitters. What is preventing them from like messing up?
Okay, that's a very good question. The reason why it's very good is we still are debating the answer for that question. So the question is, well, I have said there are hundreds of neurons, thousands of neurons in the same area, but her question is, if there are thousands of neurons in the same area, how can they communicate specifically, and what happens if one neuron is trying to release its neurotransmitters? What prevents it from reaching all the neurons around it? So there are multiple things that happens. So a synapse is an indication of one-to-one signaling. So you do not have neurotransmitters that released in a synapse diffusing out. So there are mechanisms that prevent it. So there are cells called glial cells which end up taking up whatever neurotransmitter is released, so that it is not just free to diffuse around in the extracellular space. So that's one answer, but there are multiple answers for that question. And whenever you screw these things up, you end up getting diseases. Okay? Yes? Chinese what?
Chinese whisper.
I do not know what that is.
Like when you're transferring information from one person to another, they eventually messes up.
Yes.
But then these neurons, they're able to effectively transfer information from A to B across thousands more.
No, see, there are multiple things that are happening here, right? When somebody is hearing what you are saying, they are first assuming you are saying something, right? So it's not just what you hear. It also has to do with what you're expecting to hear. So, but that is a much higher level processing compared to what we are seeing. What we are seeing is very simple. We are just looking at communication across two neurons, which is happening in a micron space. Okay. Okay, so what we do usually is what you're seeing here is a retina. You sacrifice the animal, we usually work on mice. Sacrifice the animal, you take the retina out, you lay it flat, this is what it will look like, and then you put it under the microscope, and in fact, when you see the cells, this is what it would look like. In this case, there were cells which were fluorescently labeled, some specific cell types, and so you can see the blue tag which is present only on some cells. So we know that is one cell type. When we are recording from that neuron, we know what its function will be. Okay, so that's one. But as you can see, these small structures, you can see circular ones, that is the soma. Okay, and you can see this sort of line that is coming out, that is the electrode. That is the tip size of the electrode. Okay, so what this would look like, forget this one. So for example, this is a microscope, this is a video of showing how this recording would look like. So what you're seeing here is a blood vessel. You lost that. So you can see that this is a blood vessel, these are RBCs. Okay, they are moving in there. And so now you can see this circular structure and you can see this pipette. So eventually what we end up doing is we will put the pipette on the cell so that we will record the activity coming from only this cell. As you can see, there are so many cells surrounding that, but glass is a great dielectric, and so it does not conduct. So only the signal that is going through the tip ends up being read. Okay? And so, for example, if you do that, and if you were to record the activity from, say, ganglion cells, you get very different types of responses for the same stimulus. In this example, what I'm showing you is, so the line on top is an indication of when the light is turned on. Okay, so here the light is off, then you turn on the light, then you turn off the light. Okay, so we are showing this to the whole retina, and you're recording from different neurons. And you can see that one type of neuron, it responds when the light is turned on. It does not respond when the light is turned off. Okay. If you look at this second type of neuron, it does not respond when the light is turned on, but it responds only when the light is turned off. Okay. There's a third type of neuron here, which responds when the light is turned on, and it responds when the light is turned off. So this is now three types of ganglion cells I have shown you here. Okay. And so you can, now this is a very simple stimulus. As we complicate the stimulus, it becomes evident that there are many more types of cells, and as I said, at the latest count, we think there are at least 50 different types of ganglion cells in the mouse retina itself. Okay, so again, you record from individual neurons and you look at their activity, and you can see when you give different patterns of light, the response to these patterns of light is very distinct. That is how we distinguish the cell types. Okay, any questions on that idea? Yes?
So we realize that the information is decomposed in different things.
Correct. Okay, so this is how we know the same information that you see. The stimulus is the same for all these three cell types, right? But each of the cell types seems to be picking up a different aspect of the information. The information is light went on and light went off in this case. Okay, you can have situations where what is the pattern of light? This is very simple. You just turn on the light, you turn off the light. But what happens if the light is actually a spot? What happens if the light is a different shape? Those are different aspects that are carried out by different cell types. You have to also remember, or I have to tell you, that not all processing happens at the level of the retina. The retina does some amount of processing, but higher levels in the brain end up doing much more complicated processing. Okay? So, again, according to our latest count, so that was just 3, if you look at the physiology or the electrical signals, we can find out that there are at least 50 types of cells based on the way they respond to the patterns of light. And as we pointed out, that is how we know how many different types of cells there are. So this is one question, how many different types of cells there are. What do these cells do? What is the aspect of information that they are picking up? And how are they doing it? Right? So the other point is, well, I have 50 types of cells. How are these cells? What are these cells connected to that allows them to extract these distinct pieces of information, right? So for example, these are all ganglion cells. Why are some ganglion cells able to respond when the light is turned on and not when light is turned off? Why are some ganglion cells able to respond when light is only turned off, not when it's turned on? Why are some able to respond to both, right? So these are the kind of questions that we are grappling with. What I'm showing you is a very simple example. We already know very well why these differences are, but the question gets more and more complicated. Okay, so that's the overall idea. So let's see what else I have. Okay, so I want to give you a flavor of one type of cell. So among the 50, there are at least 4 or 5 types that are called direction selective ganglion cells. So what they mean is that, for example, if you record from a direction selective ganglion cell, and if I were to now, I'm not just turning on light or turning off light. I'm actually showing it moving objects. Okay? If I were to move an object in one direction, the cell will respond vigorously. I take the same object, I've not changed anything. It's the same object, same size, same shape, same color pattern, whatever. But I'm just moving it in the opposite direction now. When I move it in the opposite direction, this neuron does not fire. Okay, so this particular type of neuron does not care for the shape, size, color, whatever. All it cares is for motion. Okay, so that is another aspect of the system. So this particular type of neuron looks at movement. There are some types of neurons that look at orientation, meaning if I show it a bar like this, it will respond well, but if I show a bar like this, it will not respond well. Okay, so this is the kind of information that is processed at the level of the retina. Now, as you go higher and higher in the brain, you get much more complicated processing. There are neurons which will respond only when you show faces. There are neurons, so you can think of it as you start with simple bars, then eventually, if you want to think of a hand, the hand would be how many bars? It's 1, 2, 3, 4, 5 bars arranged in a certain way. So if you're able to get that, then there will be cells in the brain. When you show a hand, they would respond. If you show them 5 bars, they will not respond. It will only respond when you have a hand. So bottom line is, information gets decomposed. You get these simple, from the visual scene, information gets decomposed into smaller bits. And in this case, I've given you an example, couple of examples. One was when the light turns on, or light turns off. Very simple examples. And in this case, I'm showing you where the cell is looking at only movement. Now, as I said, there are 40 other channels, right? So there are 40 other channels, and they're all doing something different. And eventually, what it allows you is it allows cells higher in the brain to use this information to come up with much more complex processing. For example, like looking at a face. Okay? Okay, so one last point I want to get across is, as I said, we are interested in not only understanding what are the differences in these cells, we also want to understand how these differences are being brought about. What are the cells they are connected to, and what is the role of the cells that are connecting that leads to these differences. So in this case, what we have to do is we have to either turn off or turn on one specific subtype. I told you there are 100 types, right? If I don't want to activate all of them or turn off all of them, I want to activate or turn off only one of these subtypes, and that is possible now. So we have a technique that is called as optogenetics, where you can express a protein called channel rhodopsin. You don't need to know the details, you just need to get an idea of the understanding of what is possible, right? So you have these proteins expressed only in specific types. For example, if you're looking at at 15 types of bipolar cells, maybe this method can be used to label only one type of bipolar cell. And then what you can do is you can directly activate them using light. These channels are light sensitive. If you shine light on the cell now, and if they express that channel, they are going to now get excited directly. And what that allows you to do is, if I were to record from this neuron, let's say, and I want to know if it is connected to these subtype of neurons among the 100 types, we can use this method to evaluate whether they are connecting or not. And by that logic, if you want to understand if some neuron is important for some computation, for example, I showed you something called as direction selectivity, where a neuron responds when an object moves this way, but not when an object moves the opposite way. If you want to understand there is some particular neuron that is important for this, then you can turn off that neuron using this method and then see if that affects your computation. Okay? Yes? So let's leave it. So how do you turn off the neuron? And then this will be the last slide. So the way we would turn off a neuron, so how does a neuron get turned on? No, so the way we turn on neurons usually under natural conditions is by electrical pulses, right? So you just have to come up with a way that will prevent electrical pulses from happening. And so what you end up doing is we do something called as hyperpolarize the neuron, meaning we reduce the membrane voltage of the neuron, so when a pulse comes in, it will not transmit that signal. Okay, so that's the way it is done. And that is possible now. Okay, so again, the idea, so I just want to give you a flavor of what are all possible. So I've given you sort of two tools. The main one was electrophysiology, where you can record the activity of one neuron from the whole network. The second was again optogenetics, where you can use this to activate one subset of neurons in the whole tissue. We also have other methods where we can look at activity of neuronal populations using calcium imaging. Again, I don't think I will be able to touch on any of this. If you're interested, you're welcome to read up on this. Calcium imaging has come up in a big way. We also have other methods where we can directly monitor neurotransmitters. So we have dyes when it's expressed in neurons, and when neurotransmitters are released, it is going to bind to these dyes and it's going to change fluorescence. So there's a lot happening in this field in terms of trying to understand how neurons process information. And I want you all to just, I don't want it to be daunting. That's the bottom line. I want you to just have a flavor of what is possible and how far we have come in trying to understand how neuronal circuits function. Okay? All right, any questions on this? And then we will stop. Any questions in general about neuroscience? Yes?
Not sure how accurate my information is. So if I did here, to even accurately model 1 gram of the brain, you would need terabytes of information.
See, again, we have to remember modeling a gram of a brain means you have to understand there are hundreds of neurons, different types of neurons, they're all connected to each other. So it doesn't matter whether you're modeling 1 gram or 10 grams. The complexity of the information that is in there is already high. So if you're able to model 1 gram, you're likely to be able to model 10 grams. So because that is scalable. But the bottom line is it happens at a really small scale, the diversity. And that is the problem. We still don't know.
How do you model the brain?
We don't know at this point. Right? We're still struggling with this idea.
To study specific for past brain researchers like grow brains in labs, right?
So this idea of, it's called organ on a chip, that's called now. So what they do now is you can, using, say for example, human stem cells, you can end up, you can program them to differentiate in a very, in a manner you want to do it, and you can get some aspects of it. For example, nowadays, it is very expensive first, but having said that, the latest in the retina, at least, you can create it, you can get layers, you can get these neurons, but they do not connect the way they should. And so that's the bottom line. There's so much more happening in there. So now the question you're asking is sort of, how does this connectivity develop? Right? So development is a huge deal, obviously, because if you're having 100 different types of cells, they all have to know who to connect to, how to go about it. And so yes, to some extent it is possible, but we are not there yet, where we can just, like they show in movies, where you can just grow and then replace an organ. Yeah. Harness. What do you mean? You want to use that as energy?
I don't know, store it. If a person dies,
Hmm.
does that mean that their neurons
Morbid.
immediately die? Or is it energy?
Neurons immediately die. What do you mean?
Yeah.
So,
Within that 7 minutes,
Hmm.
is it possible to get at least a minimal amount of energy from electricity from that?
okay, see, first of all, you have to remember. So, see, I think the important point here is the brain is a highly efficient system, meaning the amount of energy it uses to do its job is very low. So, if you're going to actually go back and get the energy that it is using, it will be minuscule. That's the bottom line. If you are going to use a supercomputer, that is going to use a lot of energy, whereas the brain uses very little energy to do the same.
Also, actually, sorry, this program, sir, we had a lecture talking about
Hmm.
the using a biological system to build an efficient computer.
Ah,
And sir, like the brain is actually really efficient.
Correct.
So, how efficient could a computer be? Can we build use a brain to build a computer, sir? Because for computers, you need to tell it.
Hmm.
You need to tell it how to process the information.
I mean, I think the first problem is we do not understand the architecture of the brain. If we can replicate the architecture of the brain, then perhaps we will be able to understand how. See, again, the bottom line is there are two questions here. First is, can we achieve what the brain achieves? Forget about the amount of energy that is used. Yes, the brain is a very efficient system, but can we even at least achieve what the brain does, even using large amounts of energy? Even that we are not there yet. If to actually come up with a system where you can use the amount of energy that the brain does to do the same amount of work, we are nowhere there. Hmm. Yes.
We take a direction selective neuron
Hmm.
and it provides stimulus in the opposite direction.
Hmm.
Isn't that essentially turning off the neuron?
Correct.
Then why do we need to prevent electrical impulses when we can just...
How are you turning off the neuron? So I guess I'm confused. So what was your question again?
Sir, when we say we turn off the neuron,
Ah.
basically the neuron's not giving us a response.
Yes. Why is that happening? See, photoreceptors are getting activated, bipolars are getting activated. So normally the ganglion cell should get activated, but it's not getting activated. Why? By some other mechanism. Okay, yeah. So it's not like the photoreceptors are not getting active, because there are cells which are not direction selective. Okay, so if you look at two cells, one, if I show a moving bar of light, one cell will respond, the other cell will not respond. That's the bottom line. Okay, they are both receiving inputs from photoreceptors. They are both receiving inputs from bipolar cells. Okay. Yes. Why do they have to be? Their question is, what is direction selectivity's importance to animal behavior? Okay. So now, if I have an object that is moving in a direction, what do? No, you don't run away, right? I have a ball that is going across my view. What do? So how do you focus on it? So you need to know which direction it's moving in, right? So only then you will be able to focus on it. Motion is a very huge field. We are very, for us at least, stationary objects, we are very poor in differentiating from the background. The moment it starts moving, we can pick it out. So we have a huge amount of brain space that is allocated for motion processing. More for praise, actually. Okay. Any other questions? Yes?
I have a weird question.
Sure.
So, like I said, this is the cell that detects direction separately. So, if you turn the cell upside down, would it sense direction in the opposite?
So that depends. See, when you say turning it upside down, the neuron is not easily turned upside down because it is connected to other neurons in the network. If you were to invert the whole network, yes.
Okay, then, so if obviously one neuron is sensing direction in motion in this way, there should be other neurons that sense direction in the opposite.
That is correct. So as of now in the retina, we have neurons that sense direction in 4 directions, the cardinal. There's one that senses north, one south, east and west. 4 types.
Can you model one neuron as like the mirror image of the other neuron?
That is what we do now, yes. Yeah. So the basic connectivity pattern of these 4 neurons is similar. It is just that, as you pointed out, they are sort of rotated. Okay. And the connectivity pattern is important here. So again, the bottom line is, you study one type, you will be able to understand how this works. For example, I'm just giving an example that happens in the retina. If you look at higher in the brain, again direction selectivity is generated irrespective of what happens in the retina. So again, the models are similar, as you pointed out. If you were to rotate that network, again, you would see that. And direction selectivity is not specific to vision. For example, if you have something crawling on your hand, it's not enough if it's just standing. If it's crawling, then it's all the more. So then you want to know which direction is crawling, so that you do that, right? So even audio, everything. Direction selectivity is something that is universal to sensory system. In fact, you can think of it even in the form of words, right? If you look at a certain series of words that happen in a sequence, in time, then you can think that's the word that you're thinking of. THE is the, right? So that's the sequence. ETH is the opposite sequence. So that is not something that triggers for you, right? Okay, kids? Enjoy, all the best. Any other question you can ask me now. I'm also available on email. Yes?
What interesting research has been going on in your department, sir?
In my department? Oh my God. What interesting research?
What has been happening?
Everything is interesting. It depends on what your interest is.
I'm interested in the way you can like the brain, sir. So has anything been done? I mean, compute like the brain.
Compute like the brain. We have somebody who works on computational neuroscience. You are welcome to contact him. Professor Srinivasa Chakravarti works on. There is a brain center. Brain center is different. I don't know what that means. What do you mean brain center is different? What happens in the brain center? They are working on reconstructing the brain. So basically they are imaging, they're cutting the brain into sections, they're imaging it. So you guys get to go there at some point? Then what are you asking me? You should be telling me. Oh, they just showed you outside.
They told they have some literature on it.
So they're reconstructing it.
How do you like? For example, if I first time I throw a ball, I'm not sure which path it will take. But when
Why?
I throw it,
What do you mean?
Like in the first time in life when I throw a ball, like in the direction sense, I might not know exactly where it will land. My aim and all might not be very visual. But then as you throw it again and again and again, you get a sense that you can predict where it goes.
Okay.
How does that prediction?
So basically, now there are 2 aspects to it. See, one is prediction is inherent to the projectile. That has nothing to do with memory. The second question you're asking is, if I do it multiple times, then I sort of know what is going to happen. So that has to do with memory. So you have enough experience of the same thing, it gets stored in memory and then it gets retrieved when that particular sequence of events happen, when you see a ball flying towards in a particular projectile. So again, memory is a huge part of what we see or what we perceive. Okay, yes.
Swayam Prabha. Digital India. Educated India.